Hey everyone. So today we have an Excel spreadsheet that's set up for you so that we can look a little bit more at what this idea of Hardy-Weinberg equilibrium is all about. So we have some cool Excel things to do, and we have some more practice doing graphs. Thinking about the idea of what does it mean when a population is in Hardy-Weinberg equilibrium. And I'm also going to show you how to do the chi-square test in Excel. All right, so this worksheet, this activity doesn't take very long, but it seems a little bit complicated and confusing when you first look at it. So I'm going to walk you through the steps. All right, so the idea of Hardy-Weinberg equilibrium is that if we have a large randomly mating population, the allele frequencies of the offspring should equal the allele frequencies of the parents. So if nothing is happening to change those allele frequencies, which would be evolution typically, then what we should have is a population of offspring that matches the allele frequencies of the parents. So this is a little boring because we're going to get a result that nothing is happening, nothing is going on. But we can really see what Hardy-Weinberg is all about from the simulation. All right, so we're starting out with allele frequencies, the top left-hand corner up here. So if you look at these values for little p and for little q. So if we want to change these values, we just have to change it in the first box for little p, because the equation is set up in box C4 there to take 1 minus little p to equal q. All right, so if you noticed, everything changed all the way down the spreadsheet. When I made that change, I'm going to change it back. All right, so what happens when we change those allele frequencies? What are those allele frequencies being used for? All right, so we are creating a population of 1,000 individuals. That's a pretty large population. If you scroll down all the way, this just keeps going and going and going and going down here. Right, so we are making 1,000 individuals, and we are creating genotypes for each one based on randomly picking 2 alleles to put together into the parent's genotype. These 2 alleles, big A1 and big A2, have whatever allele frequencies we have up here. So this function, if you look up at the top at this function, it has these rand values. So this is a function in Excel that picks a random number. In this case, where it picks numbers with digits, decimal places, and if it's bigger than 0.5, it's going to make it an A1, I think, and if it's less than 0.5, it's going to make it an A2. So we just have set up a way to decide using random number generator which of the 2 alleles are going to be used to make up the genotype. All right, so then for each parent, we're going to pick one of those alleles to make a gamete. And so this individual 0, it's an A1 individual, so its gamete is going to be A1. This is an A1, A2 individual. It could have a gamete that was either A1 or A2. We have randomly selected A1 in this case. All right, so now we're going to make a population of offspring based on these gametes. And this simulation assumes that any individual could actually be a mom or a dad. All right, so just like we did, we picked random numbers for us to sample beans out of our population. We can do a similar thing in Excel that we use that randomizer.org website to do. We are going to make a population of offspring that's also 1,000 individuals by first picking a mom at random, and then this next cell is going to pick the gamete that that particular mom has. All right, we're also going to pick a dad at random, and write down the sperm that the dad contributed based on what's in column C over here. And then we're going to make a genotype for the offspring by combining together the egg and the sperm. All right, and you can see the formulas for all of these steps in the, if you look at the formula bar at the top. All right, so we've made these offspring. And over here on the right-hand side, there is a set of calculations that we're doing for the parental population, right? So these are going to be counting up. This is a nice little function, count if. This is going to count up how many A1 individuals ended up in my parental population, how many heterozygotes, and how many homozygotes. Right, so we're going to do a quick check as well to see if our parental population is in Hardy-Weinberg equilibrium. It should be, right? So there's a few little formulas going down here. Notice what the last one says. I found a p-value here, and our null hypothesis is that the population is in Hardy-Weinberg equilibrium. Right, so if we don't reject the null, the answer to the question, is this population significantly different from Hardy-Weinberg prediction? All right, we don't reject the null, the answer is going to be no. All right, so if this says no, that means my population is in Hardy-Weinberg. That should be true. All right, so if you change a value up here in cell C3, then it's going to refresh the whole spreadsheet. But you can also refresh the whole spreadsheet and get a completely different set of random individuals by hitting the F9 key. All right, F9 is basically like hitting enter again. So you can see that these values change a little bit. Every time I do it, I'm going to hit it again. All right, we still get this answer of no. All right. Now, we've done a little bit more randomizing, moving from the parents to the offspring. So I've highlighted the offspring population over here. And what you're going to see is, again, we've tallied up how many individuals we have that are each of 3 different genotypes. And then we also are going to say whether we ended up with a population of offspring that was significantly different from Hardy-Weinberg. Right, so in this case, we are looking at a calculation of the expected values. Where are we getting that from K11. So we're using our expecteds for the offspring population is actually the ones that we calculated for the parents. All right, so we're comparing our parents to our offspring in this Hardy-Weinberg test. Okay, all right, so there's a few things the worksheet is going to walk you through. And kind of testing out how this this spreadsheet works and thinking about what does it mean to be in Hardy-Weinberg equilibrium. All right, so that's our null hypothesis. So our population should be. Occasionally, you will get a population that's out of Hardy-Weinberg equilibrium, even though it should be. Right, so it's possible that you can get a yes in this answer here. If you keep clicking through, you'll occasionally get a yes for this answer, but usually you shouldn't get it very often. Right? You shouldn't get it very often. How often you get it should be set at our 0.05. So 5% of the time out of all the trials that you did, that's what our alpha level is going to set. All right. So there's a couple interesting things we can do with this spreadsheet once we have it up. And I'm going to ask you to do a little sensitivity analysis or a little simulation also over here on this side. So when we have our allele frequencies at 0.5 and 0.5, we have a result of our expected genotype frequencies that we're going to put in here. We're going to copy, and this was 0.5 and 0.5. So we're going to paste special values. We're going to put those numbers in over here. Right, and so those are expected genotypes if our allele frequency is 0.5. Now, what we're going to do, this little table is set up for you to systematically go through and change the value that's in cell C3 over here. And so our frequency of A1, now we're going to change it to 0. And when I click enter, I get, again, a whole bunch of different values here. And when I go over here to my expecteds, again, I'm going to copy this and I'm going to paste it into values into this table. Now I'm going to change it to 0.1. And I'm going to, again, copy and paste these values using paste special over here. Right, once I get my table all filled in, I just want you to make a graph. You're going to highlight all of the cells here and then go to insert. And we're going to do a line graph. We've got multiple lines with markers, right? It's not finished. It's going to give you, I'm going to move this legend. Oops. Over to the right. Right, we want to add an axis label on the y-axis. I'm going to use chart element, axis titles. Now what I'm putting on this horizontal axis, what are these numbers? Those are the frequency of the A1 allele. Right, I also need an axis title for my vertical axis. That is my expected phenotype frequency. All right, out of 1,000. So it's a count or not a relative frequency, but an absolute frequency. Right, and so you can see as I'm filling in this table, the rest of my graph will fill in. So then I want you just to describe what pattern you're seeing on this graph so you can finish that. Okay, oops, I didn't want to do that. All right, now we're going to do a chi-square test using Excel as well. And there's a couple different ways you can do it. You can actually type in the equation for the chi-square test and use Excel as a calculator for you, but I'm going to show you how to do it for any data that you want to use. And what you're going to do is make sure you have a row in your table. This is the example from lecture, that you have a row in your table that's your observed values, or you have a whole table somewhere that's your observed values, and you have a row or a table that's your expected values. You need to type into a cell somewhere. You have to, the thinking scientist, you has to tell it how many degrees of freedom you have. So for all of these genetics examples, we're going to have 1 degree of freedom. If we have one gene with 2 alleles, we're going to have 1 degree of freedom. So we're going to get the p-value for this test using a function. All right, we don't need to go to data analysis tool pack. We're just going to use a function. We can type it into any cell, and we are going to use chi-square test, CHISQ dot test, and it tells us to put in the actual range, comma, the expected range, right? So that's the observed cell range and the expected cell range. We can do this for goodness of fit tests. We can do this for independence tests. We can do it for really big tables. It doesn't have to just be one row, right? So if our actual range is not a 2x3 table, but it's a 4x5 table, we can do that too, right? We're going to close parentheses. We're going to get our p-value. All right. And then we also want to report our chi-square value. Now, this is backwards from how we would do it by hand, right? If we were doing this by hand, we'd need the observed and the expected, and we'd calculate the chi-square value. Doing it in Excel, we're going to get the p-value first. Right now, we're going to do equals chi SQ .inv, the inverse of the distribution, RT, the right tail. That's the test we want to do. And it says we need to put in a probability and a degrees of freedom. So probability, comma, degrees of freedom. And that's going to give us our value of chi-square. All right, it's very small. That means it's very close to the left-hand end. That's why we got a really big p-value. And we also, if we want to look at how different our observed and our expected counts are, we can make a quick graph of that as well. And so I want you to make a graph of some of your data for a chi-square test in the worksheet. You'll do it exactly the same as this. This is one of the easiest graphs to make. We're going to go to column chart, and we're going to do the side by side. All right, so this is a really boring looking graph because our observed and our expected are really similar to one another. Right? So we see that when we actually graph them, they're so similar that we really wouldn't expect to see that we had a difference between the observed and the expected. Right, so for this example, our population is, we have a p-value of 0.98. We do not reject the null. We see from our graph that we have observed and expected values that are not very different from one another. We shouldn't be rejecting the null here. And I'm going to finish this graph by adding my axis titles. My horizontal is genotype. And my vertical is all right, so those are the different skills that we're going to do in Excel associated with Hardy-Weinberg equilibrium and an introduction to using chi-square test in Excel. And the genetics worksheet, again, there's a whole lot here, but the purpose of it, don't get overwhelmed with all the details. We won't make you do anything like this. Again, it's just a way that we can use Excel in order to draw actual random sample from a population and look at what a population in Hardy-Weinberg equilibrium should look like.